Hello, welcome everybody to Trailhead's second webinar of 2025. My name is Dr. Angelica Gomes Iotti, and I am a scientific director here at Trailhead Biosystems. Before we begin, I would like to take a moment to let everyone know that today's webinar is being recorded and will be available shortly on our website. All attendee microphones have been disabled for this presentation. We will have a short questions and answers session at the end of today's presentation. You can submit your questions to our team through the chat question submission button in your team's navigation bar, and we will try to get through as many questions as possible within our time. If you have any questions after the live webinar is over, you can reach out directly to our team at support@trailbio.com. Today, I have the privilege to introduce Dr. Michael Elias, our field application scientist at Trailhead Biosystems, who will be presenting today's topic, iPSC-Derived HPCs, enabling scalable and standardized blood cell production. Dr. Michael Elias earned his PhD in nanoscience from UNC Greensboro, where he focused on stem cell differentiation for cancer therapy. He's a stem cell biologist with expertise in regenerative medicine, immunology, and translational research. After completing postdoctoral work at Cleveland Clinic, where he studied inflammatory diseases and fibrosis, he joined Trailhead Biosystems, where he has helped develop and scale protocols for iPSC-derived hematopoietic progenitor cells over the past 4 years. His work bridges cutting-edge cell biology with real-world therapeutic applications. Michael, thank you for presenting this webinar today.
Okay, thank you, Angelica, for the beautiful introduction. Let's just kick it right off then. So here's a quick overview of what we'll cover today. First, I'll introduce Trailhead Biosystems and then touch on hematopoiesis as the foundation of blood development. Next, we'll look at why iPSC-derived HPCs are valuable compared to donor sources. I'll then walk through Trailhead's approach, the iPSC-derived HPCs that we developed at Trailhead using our HD-DOE platform. And finally, we'll wrap it up with key applications on these cells. Trailhead Biosystems, founded in 2015 and based in Ohio, focuses on generating specialized cells derived from the mesoderm, endoderm, and ectoderm. Pictured here is our facility. We are located in Beechwood, Ohio, just outside of Cleveland. Turn the laser pointer on. Since our mission is to create specialized cells from stem cells, it's important to highlight where the real challenges in the field lie. Many years of research have gone to reprogramming iPSCs, and now we know how to reliably bring them back to a versatile state where they can become any cell type. The real hurdle isn't reprogramming, it's guiding these iPSCs into the right specialized lineages. That's where Trailhead comes in. Our work focuses on solving this differentiation challenge using iPSCs as a customizable platform to generate functional, specialized cells. These cells can be applied across multiple areas, from cell therapy and drug discovery to disease modeling and tissue repair. Let's talk about the major challenges with iPSC-derived cells today. First, the demand for high-quality human cells is enormous, but right now, the options are limited. Researchers and companies alike are eager to use these cells for therapy, disease modeling, and drug discovery, but the field continues to run into the same problems. Second, obtaining pure populations can be a challenge. Many of the available cells simply don't meet the standards needed for consistent and reliable results. Also, there's a lack of consistency from batch to batch. That means a researcher might not get the same set of results for one vial, and then also just get a completely different set of results from a different vial. Fourth, supply is a bottleneck. These cells just aren't available in the quantities the market truly demands. And finally, much of the development production is still being done manually. This slows down everything and introduces opportunities for error, which make it very difficult to scale. Together, these challenges create a major roadblock for the field, and solving them is the key to unlocking full potential of iPSC-derived cells. Now that we've looked at the problems, let's move into how we can solve them. At Trailhead, our approach is to fundamentally change how stem cell discovery and manufacturing are done. Rather than relying on slow, traditional, iterative processes, we leverage machine enablement to accelerate discovery. This allows us to test and optimize conditions at a scale that simply isn't possible by hand. We also use this approach to efficiently explore the high-dimensional space of regulatory inputs, the combination of growth factors, small molecules, and culture conditions that control cell fate. By doing this, we can fine-tune the differentiation pathways in a way that drives both precision and consistency. The result is vastly improved cell purity and the ability to generate specialized subtypes that meet the needs of researchers and clinicians. With identified critical process parameters, these cells then can be manufactured reproducibly and at scale. And because this entire system is built on a flexible production platform, it can be applied to multiple cell types while remaining cost-effective. The result is broader access, more reliable results, and ultimately real impact for patients and in the whole field. So how do we actually make all this possible? The answer is our HD-DOE platform. HD-DOE stands for High Dimensional Design of Experiments, and it's the core technology that drives everything we do at Trailhead. Instead of relying on trial and error, we use proprietary software tools that we've developed in-house to design experiments at scale and complexity that would otherwise be difficult to achieve. These experiments are then executed using computerized robotic systems, which means we can achieve a remarkable level of speed, precision, and scalability. All the data generated through these experiments is novel, giving us a unique and expanding knowledge base. And because everything is data-driven, we can determine critical process parameters in an unbiased, empirical way. The combination of advanced software, robotics, and data ownership is what allows us to exponentially accelerate cell protocol development and reliably generate specialized cell types. So here's a visual example of our HD-DOE platform. If we look at 12 factors and how they affect cells, traditionally that would require about 4,100 experiments. With HD-DOE, we can compress that large, resource-intensive 4,100 experiments to just 96, collecting the same amount of data, modeling that data, and predicting the cell fate outcome. This allows us to generate a lot of robust and reproducible protocols at an incredible pace. So let's move into the background of hematopoiesis. To understand the foundation of blood development, we need to start with hematopoietic stem cells, or HSCs, the rare population of cells at the very top of the blood hierarchy. HSCs are unique because they're the only cells that can enable long-term and secondary engraftment. In other words, they don't just reconstitute the blood system once, they can sustain blood production for a very long time. They also have the ability to generate any blood lineage. From red cells and platelets to myeloid and lymphoid lineages, HSCs again sit very top of the hierarchy. A critical property of HSCs is their self-renewal capacity. This allows them to persist and maintain stem cell pools within the specialized environment of the bone marrow. However, it's important to remember that HSCs come with significant challenges. Outside the bone marrow niche, they are difficult to expand and maintain while preserving stemness. This is one of the biggest barriers in translating HSC biology into scalable therapies. Finally, long-term HSCs, which is the population found in the bone marrow with self-renewal capacity, can be identified by a well-defined set of surface markers, including CD34 and CD90 positive, CD45RA, and CD38 negative. These markers are used in both research and clinical contexts to distinguish HSCs from progenitor populations. So, in conclusion, hematopoietic stem cells represent the gold standard for long-term blood formation, but they also illustrate why in vitro differentiation approaches like the ones we've discussed are so important. They give us tools to study, model, and eventually harness HSC biology in a way that overcomes the limitations of donor-derived materials. Now that we've looked at the unique properties of HSCs, let's look at how hematopoiesis happens during development. There are two major processes: primitive hematopoiesis and definitive hematopoiesis. The first hematopoietic progenitor cells, or HPCs, emerge during primitive hematopoiesis, which is extra-embryonic and takes place in the yolk sac. This wave mainly generates red blood cells along with early erythroid and myeloid progenitors, providing oxygenation and temporary blood support for the growing embryo. Later, within the embryo itself, we see the onset of definitive hematopoiesis. This begins in the aorta gonad mesonephros region, or the AGM, where hemogenic endothelium gives rise to the first hematopoietic stem cells, or HSCs. HSCs emerge into the bloodstream and migrate to the fetal liver, where they expand, mature, developing a distinct transcriptomic profile. As development continues, HSCs move into the bone marrow, which becomes their lifelong home. In the marrow, they stay in a quiescent state under hypoxic conditions. When the body needs new cells, these HSCs are activated and differentiate into a full range of hematopoietic lineages. So, overall, hematopoiesis begins with a short-lived primitive wave in the yolk sac, but then the definitive wave in the AGM is what establishes HSCs that persist for life. In the embryo, the first long-term hematopoietic stem and progenitor cells, or HSPCs, arise in the AGM. Within the ventral wall of the dorsal aorta, a specialized group of endothelial cells called hemogenic endothelium develops the capacity to generate blood cells. During a process known as endothelial to hematopoietic transition, these hemogenic endothelial cells begin to change identity. They maintain endothelial markers such as VE-cadherin and CDH5, while at the same time switching on hematopoietic transcription factors like RUNX1. Along with the transcriptomic reprogramming, the cells undergo a striking change in morphology. They round up, loosen the tight junctions that hold them into the vessel wall, and eventually detach. As they pull away from the endothelium, they butt into the aortic lumen and form what are known as the intra-aortic hematopoietic clusters. The cells in the clusters are newly specified progenitors. They can be identified by surface markers such as CD34 and CD43. More importantly, they gain the ability to generate multiple blood lineages. The AGM-derived HSPCs produced by this process are the very first cell with true long-term self-renewal and multi-lineage potential. These cells go on to seed other hematopoietic sites, first the fetal liver, and eventually the bone marrow, where they will establish the foundation of the lifelong blood system. So, why iPSC-derived HPCs? On this slide, I want to emphasize where hematopoietic progenitor cells are typically sourced from and why these sources are limiting. First, the three major donor sources are cord blood, bone marrow, and mobilized peripheral blood. Each of these has been critical for both research and clinical use for decades, but all of them present challenges. Cord blood is limited by the volume that can be collected per birth. That means the number of cells per unit is small, and there's a lot of variability in yield and quality between different units. Even with cord blood banks, the supply is ultimately finite. Bone marrow is a more traditional source, but collection is invasive for donors, and it isn't always easy to find a suitable match. The quality and expansion capacity of the cells can also vary from donor to donor. Mobilized peripheral blood requires treating donors with mobilizing agents to move stem and progenitor cells out of the bone marrow into the bloodstream. Not all donors respond the same way, and the mobilization process itself adds cost, complexity, and regulatory hurdles. Another challenge with primary cells is donor compatibility. For allogenic use, immune rejection and the need for HLA matchings are constant concerns. For autologous use, sourcing patient-specific cells is limited and not always feasible, which restricts how broadly this approach can be applied. Finally, it is difficult to create a bank of edited cells. These limitations make it very hard to achieve the kind of standardization and reproducibility that modern research and industry currently demand. Induced pluripotent stem cells address many of the shortcomings we see with donor-derived HPCs. First, they bring consistency. Because iPSCs can be banked and used as starting material, differentiation runs can be repeated under controlled protocols. This minimizes lot-to-lot variability, which is a major issue when working with donor material where every lot can behave differently. Second, iPSCs allow for customization. You can generate lines from specific patients or even engineer them to model a genetic disease. This opens the door to disease-specific applications. Third, they provide an unlimited source of cells. Unlike cord blood or bone marrow, which are, again, finite, iPSCs can be expanded indefinitely in culture. This ensures the supply of HPCs is renewable and scalable to any level of demand. Another key benefit is reproducibility. With standardized manufacturing, iPSC-derived HPCs provide consistent populations that make data comparable across experiments, labs, and even across institutions. This is critical for advancing both research and preclinical pipelines. iPSCs are also scalable. They can adapt to large-scale production systems, making them useful not only in the research lab, but also in industrial platforms, high-throughput drug screening, and preclinical testing. Finally, the applications are broad. iPSC-derived HPCs can be used to study hematopoietic differentiation, model blood cancers, bone marrow failure syndromes, autoimmune disorders, test for drug toxicity, and explore immunology through differentiation into downstream cell types like T cells, NK cells, or myeloid cells themselves. In regenerative medicine, they represent a potential source of therapeutic development. Altogether, iPSC-derived HPCs provide a consistent, reproducible, and scalable solution to many of the barriers faced within donor-derived sources. On this slide, we're looking at donor-derived HPCs compared with iPSC-derived HPCs. For donor-derived HPCs, the biggest limitation is availability. Whether we're talking about cord blood, bone marrow, or mobilized peripheral blood, the supply is finite and often hard to scale. There's also high donor-to-donor variability, which makes reproducibility across experiments difficult. Collection itself can be invasive or logistically challenging, and on top of that, regulatory complexity adds further barriers. Another important point is that donor HPCs are hard to customize. You can't easily match them to a specific disease state or patient background. Now compare that to iPSC-derived HPCs. The first major advantage is supply. Once you establish a line, iPSCs represent an unlimited, renewable source of progenitors. They can be manufactured into standardized batches under controlled protocols, which improves reproducibility. Instead of invasive procedures, these cells are produced entirely in the lab, enabling scalable and controlled manufacturing. And importantly, they can be customized, whether it's patient-specific iPSCs, disease models, or engineered controls. One more point to highlight is maturity. Donor-derived HPCs are naturally more mature, reflecting adult hematopoietic biology, while iPSC-derived HPCs often resemble a more fetal-like state. This is an area of active research. Improving the maturation and functional capacity of iPSC-derived cells is a key step for both translational research and eventually therapeutic use. So, in summary, while donor HPCs have been foundational, their limitations make it difficult to move the field forward. iPSC-derived HPCs offer scalability, consistency, and customization, and represent the next generation of hematopoietic progenitor cell technology. So that brings us to the next question. How do we actually recreate blood from iPSCs in the lab? First, we begin with iPSCs, which at the earliest stage are pushed into the mesoderm. At this stage, you can see the cells activate genes like brochure and markers KDR, which are the hallmark of the primitive streak mesoderm. Once the mesoderm is specified, Wnt signaling plays an important role in guiding the cells towards a hemogenic fate. Additional signals then reinforce the trajectory towards the lateral plate mesoderm, which is the developmental origin of both blood and vascular lineages. As the cells progress, they form what we call the hematopoietic mesoderm, which can be identified by markers such as KDR and CD235. From there, they transition to the hemogenic endothelium, marked by CD34 positivity in the absence of CD43. This stage is pivotal. These endothelial-like cells are sitting right on the edge of becoming blood. At this point, the culture conditions guide cells toward an endothelial identity, while others adapt to hemopoietic fates. From there, the populations can branch into two programs. In the primitive programs, cells differentiate into early erythroid and myeloid lineages, supporting oxygen delivery and innate immune function during early development. In the definitive program, cells generate multipotent progenitors that can give rise to both myeloid and lymphoid lineages, and ultimately to hematopoietic stem cells with long-term repopulating potential. So, this figure gives a conceptual roadmap. Pluripotent stem cells, through cytokine cues, move stepwise through the mesoderm, hematopoietic mesoderm, and the hemogenic endothelium before splitting into the primitive and definitive hematopoietic pathways. So let's move on to the trailhead approach. Now that we've covered the biology and the background of HPCs, let's talk about how trailhead approaches the problem of generating HPCs from iPSCs. We follow a three-stage workflow. Starting with pluripotent stem cells, we first induce the mesoderm, then specify those cells into the hemogenic endothelium, and finally guide them into the hematopoietic progenitor cells. The most critical point in this workflow is stage 2, the transition into the hemogenic endothelium. This stage is the developmental gateway to blood, and getting it right is essential for producing functional HPCs. To do this, we use a proprietary HD-DOE platform. This method systematically tests and refines combinations of small molecules and proteins to fine-tune pathway activity in vitro. By controlling signaling dynamics at just the right time and dose, we can precisely direct differentiation into the hemogenic endothelium. We have screened more than 250 factors for this project. The outcome is a robust and reproducible process that mirrors embryonic development, but with the scalability and control required for research and translational use. This sets the stage for consistent, high-quality HPC production at stage 3. First, we confirmed the mesoderm identity by immunocytochemistry using two key markers, CD309, also known as the VEGF receptor 2, and the transcription factor brochure. The data isn't shown, but it gave us confidence that the mesoderm was successfully established. The next step is to now confirm progression into stage 2, the hemogenic endothelium. On this slide is stage 2, and as I mentioned earlier, it's really the developmental gateway into blood. At this stage, we expose the mesodermal cells to our optimized stage 2 media, which induces the expression of a panel of genes and markers that define the hemogenic endothelium. These include RUNX1, CD31, FLY1, and CD34. On this slide, you can see representative ICC images showing several of these markers. For example, SOC17, CD309, RUNX1, GATA2, CD31, and CD144. This confirms their expression at the protein level. We also validated this population using flow cytometry, which shows strong marker expression across the majority of cells. For example, 82% of our cells express CD31, 43% of our cells express RUNX1, and 86% of our cells express CD34. These results demonstrate the cells are not just partially transitioning, but that we're generating a robust and reproducible population of hemogenic endothelial cells. So we dove deeper and validated our stage 2 cells using bulk RNA-seq. On the right, you can see a heat map of gene expression across the workflow. The genes highlighted in red are important transcription factors for hematopoiesis, including RUNX1, SOC17, GATA2, and SPI1. These are essential regulators that mark cells with hemogenic potential. In contrast, the genes in blue are endothelial markers, such as CDH5, PCAM1, and KDR. These confirm that the cells retain an endothelial identity. What's important here is the co-expression of both gene sets. We don't just see endothelial markers or just hemogenic markers, we see them together. That transcriptional profile is the hallmark of the hemogenic endothelium, distinguishing it from the generic vascular endothelium. This confirmation is critical because hemogenic endothelium is again the developmental gateway to blood. Once established, it sets the stage for the emergence of hematopoietic progenitor cells in stage 3. To generate HPCs from stage 2 hemogenic endothelial cells, we developed a stage 3 HPC recipe designed to optimize the expression of HSC signature genes. We analyzed unsorted, thawed, trail bio HPCs, and what we see is a highly pure and early stage hematopoietic phenotype. Over 70% of the cells co-express CD34 and CD43. These are the hallmark markers of hematopoietic progenitor cells. Importantly, CD235, an erythroid differentiation marker, remains low, showing that these cells are not drifting prematurely into lineage commitment. Another critical feature is the retention of CD90. About 60% of our HPCs are CD90 and CD43 positive, and none of these co-express CD45RA, which is associated with differentiation. This indicates an early HSC-like phenotype. It's important to note that CD90 is a selective marker for early HSCs, and cells that begin differentiating quickly lose CD90. So the fact that our HPCs retain CD90 highlights their stem-like state. When we overlay the classical HSC profile, which is CD90 and CD34 positive, and CD45RA and CD38 negative, we see approximately 60% of our cells have that profile. This is a substantial enrichment and represents a higher proportion of cells with high stemness compared to what we observe with alternative commercial HPC products. So, in summary, our cells don't just express the right markers. They show a classical, early HSC phenotype that is more robustly enriched than the competitor cells, making them uniquely valuable for both research and translational applications. Now that we've established the profile of our HPCs, let's take a closer look at how our HDDO-driven trail bio recipe compares to a standard literature-based control recipe. We tested two approaches: a stage 3 control recipe, which uses commonly applied cytokine combinations, and a stage 3 trail bio HDDOE optimized recipe. The trail bio recipe was designed specifically to enrich for HSC signature genes identified by Calvanese and colleagues as defining early hematopoietic stem cell identity. To validate this, we measured expression of these genes in our cells. The Calvanese panel includes HLF, SPINK2, HOXA9, and MECOM. In batch 1, we performed RNA sequencing, which confirmed higher expression of genes in cells differentiated using a TrailBio recipe compared to the control. You can see this across all 6 genes. In order to confirm our data, we performed qPCR validation in a second batch of cells. Again, we saw significantly higher expression in cells generated with our recipe, confirming the enrichment of HSC-associated transcriptional programs. Together, these results show that the HD-DOE-driven TrailBio recipe produces cells with a stronger HSC identity compared to a cytokine stage 3 media. This bulk RNA-seq analysis compares our differentiated cells to starting iPSCs. What we see is exactly what you would hope to see in properly specified progenitors: a clear loss of pluripotency markers like NANOG and SOX2, alongside a strong induction of hematopoietic genes as RUNX1, PTPRC, and SPN. Importantly, we also observed the upregulation of HSC-associated genes including HOPX, OXA9, and HLF. Finally, these cells turn on key transcriptional factors like GATA1, TAU1, and SPY1. These are the regulators that drive lineage specification into the blood. Altogether, this profile validates our TrailBio HPCs are not partially differentiated. They're molecularly aligned with true hematopoietic commitment. Here we have our product overview. On the right, you can see bright field images of the culture showing healthy, viable cells. Flow cytometry confirms that the viability remains high, with recovery consistently stronger than what we see in competitor products. This is illustrated in the bar graph on the left, where TrailBio HPCs demonstrate excellent survival following post-thaw. Importantly, these cells display an HSC-like marker profile, which is again CD34, CD90 positive, and CD45 RA and CD38 negative. Overall, what this demonstrates is that our HPCs are not only durable post-thaw, but also maintain the characteristics needed for reliable research use. Let's move into the application sections and see how we applied these cells. To begin the application section, let's look at one of the most widely used functional assays for hematopoietic progenitors, the colony-forming unit, or the CFU assay. Here, TrailBio HPCs were seeded into methylcellulose-based media and cultured for 14 days. At the end of this period, we observed the formation of multiple distinct colony types. On the right, you can see examples: a granulocyte colony, or CFUG, a macrophage colony, or CFUM, and an erythroid burst-forming unit, or a BFUE. These represent both myeloid and erythroid lineages, demonstrating their HPCs retain broad differentiation capacity. The bar graph summarizes colony count, showing robust output across multiple lineages. In summary, TrailBio HPCs not only display expected marker profile, but they also demonstrate functional multipotency in colony-forming assays, validating their utility in downstream applications. Here, we're looking at the ability of TrailBio HPCs to commit to the monocyte lineage. After stage 4 differentiation, we analyzed cells by flow cytometry, and you can see strong populations expressing monocyte markers CD14 together with CD33. The same observation is made with CD14 and CD16. In addition to these, we also looked at other markers including CD163 and CD45. This bar graph shows that the TrailBio cells express these markers at high levels, indicating robust differentiation into monocytes. When compared side by side with competitor HPCs, you'll notice that the number of positive cells is similar across the markers shown. The data indicates our protocol produces cells with reliable commitment to the monocyte lineage. Additionally, we have conducted studies showing that these cells can differentiate into functional macrophages and dendritic cells. We found an interesting trend where our flow cytometry showed that CD14 and CD33 double positivity and CD14 and CD16 was in a higher proportion of cells compared to our competitor product. We are exploring this further, and we welcome any comments and questions during our Q&A session. This example highlights the functional utility of TrailBio HPCs. Not only can they generate multiple colony types in assays like CFU, but they can also be pushed into specific immune lineages like monocytes, opening the door for studying innate immune biology and related applications. On this slide, I want to highlight the lineage-specific differentiation potential of our TrailBio HPCs. On the left, you can see differentiation into the myeloid lineage. Our HPCs were guided through stage 4 and into myeloid progenitors, further into neutrophils at stage 5. Flow cytometry analysis confirms a robust presence of CD11b and CD15 populations, or positive populations. This is characteristic of a neutrophil identity. On the right, we have an erythroid differentiation. Here, trail bio-HPCs were directed into erythroid progenitors and then into erythroid cells. This differentiation was done in the presence of dexamethasone. Flow cytometry analysis shows that clear co-expression of CD71 and CD235, confirming erythroid identity. Together, these results demonstrate that trail bio-HPCs can be directed into multiple hematopoietic lineages, both myeloid and erythroid, providing strong functional evidence of their differentiation capacity. This flexibility is critical because it shows not only that these cells remain multipotent, but also we can fine-tune the development trajectory for a specific application. So, to wrap it up, let me highlight a few key points. First, our HD-DOE platform gives us a powerful, data-driven way to guide iPSCs into committed lineages with precision and reproducibility. Second, our iPSC-derived HPCs are scalable, customizable, and minimize lot-to-lot variation, making them a strong alternative to donor-derived sources. Third, these cells retain important stemness markers like CD90, maintain a classical HSC profile, and show low spontaneous differentiation and have a high post-thaw viability. And finally, we've shown their functional applications, forming myeloid and erythroid colonies and differentiating into multiple mature blood cell types. This combination of innovation, quality, and functionality positions trail bio-HPCs as a reliable tool for both research and translational work. So, thank you for joining today's webinar. We've covered how trail bio-HPCs are generated in their robust characterizations and their applications across multiple lineages. For more details, you can visit our website to access product information, data sheets, blogs, and FAQs. If you'd like to discuss specific applications or larger scale projects, please reach out to support@trailbio.com. Now, we will move into the Q&A with my colleague Angelica, a scientific director at Trailhead who spearheaded the scientific development of these cells and is an expert in immune biology. With that, we'd be happy to take any questions.
Perfect. Thank you, Michael, for a great presentation. We have received a few questions for the team, so we can dive straight into the Q&A. So the first question, can you talk more about the quality control and the validation testing that is performed on each batch of cells?
Hi, Tom. Thank you for the questions. We follow a bunch of different experiments to qualify our cells. First, we measure sterility and mycoplasma to make sure our cells are clean. Second, we do viability tests by flow cytometry, send our cells with cytox diet to make sure that our viability is high. As you saw, it's around 80% on the batches of cells that we have in stock. We also make sure that the clients will receive the exact cell number that is, say, on the vial, approximately 1 million cells per vial. Additionally, we measure markers by flow cytometry, such as CD34, CD43, CD90, and CD235. And we also do functional studies like colony-forming unit assay to make sure that our cells are functional.
Great, thank you. And then can you also talk about the consistency of the cells across these different lots and batches and how you measure?
Sure. Thank you for the question. So we have 4 batches in stock right now, and measuring the consistency across these batches, we are happy to say that the batches are really consistent. We use bulk RNA-seq analysis to make sure the cells have a similar profile when compared across different batches. Additionally, we do functional studies and compare the performance of the cells side by side. For instance, we measure monocyte differentiation on the cells, red blood cells differentiation, all at the same time from the different batches that we have, and the cells behave really consistently in a similar range in the tests that were performed.
Excellent, thank you. Are there plans to offer lineage-committed derivatives, for example, T cells, B cells, macrophages?
Yeah, definitely in the future, we have plans to offer specialized blood cell types. In the moment we are on the R&D phase, I would say that the most advanced protocol is for macrophage and dendritic cells. We have used our HPCs to differentiate into monocytes and then into macrophages, and also from monocytes into dendritic cells, and then G cells functional. We conduct functional tests both in the macrophages and the dendritic cells. So yeah, in the future, we are going to have specialized blood cell types in the moment we are in R&D phase.
Okay, great. And then a few technical questions. Have you been able to maintain the HPCs for any period of time post-thaw and maintain the HSC marker expression that was presented?
That is a really good question. It's a big challenge for the whole field of the HSCs, right? To expand them and maintain the staminas. I haven't done these studies yet, but I am confident to say that it is really hard to expand the cells and maintain the states. Whenever the cells are cultured in some cytokine cocktail, they start differentiating. So yeah, I haven't done that, and this is possibly a challenge for the whole field.
We have two questions that are similar. So the first is, what cell lines have you generated HPCs on? And then the related question then is whether the source iPSCs are a big factor in terms of the quality of the resulting HPCs.
So yeah, we have done differentiation of HPC using 2 cell lines that we have in-house. And the differentiation protocol that we have was robust on the 2 cell lines tested. Additionally, we have tested 2 other cell lines, being one disease, a disease-related cell line from a collaborator. So basically, he shipped the cells for us, and we conducted our differentiation protocol in-house, and we got good HPC differentiation so far with 4 different cell lines. So yeah, we are open to differentiation services in which we are going to receive your iPSC line and differentiate them to the lineage that you are looking for.
Thank you. And then a few more technical questions. Have you tested your HPCs with cancer organoids from patient samples?
Thank you for this question, since this is a really hot topic and really popular topic right now, but I haven't done that. I think it's interesting. I'm open to test that. I just haven't had the time yet.
Great. And then have you tried to engraft these cells?
This is, yeah, a really important question that, yeah, we are all looking forward for the answer. We have started some animal studies with these cells, but we are in the middle of it. We haven't got any data yet. So yeah, we are waiting for the data, and we are excited to see that.
And then the last question that we have, oh no, that was the last question. Perfect. So I think we've covered everything that has come in. So that concludes the questions for the webinar. Thank you again, Angelica, for answering the questions. Thank you, Michael, for the great presentation. If you have any additional questions, please do feel free to reach out to us at support@trailbio.com. This webinar will be hosted on the website later this week. And just thank you again for joining us today, and hope that you have a great rest of your day.
Thank you, guys.